Hello?
Can you hear the radio from?
Not really, no.
Can you hear my radio?
No.
No. Okay, good.
Yeah, I don't hear anything. A little bit, that's fine.
Oh, well, the news and the weather.
Yeah,
goes on.
yeah. Yeah. How are you doing this morning?
Am I?
Yeah,
Oh, pretty good.
good. So I got your message about I got the talk. I downloaded it, and I have it all set up here on this side. So if you want to do it on your side, if you can share your screen, then
Okay,
not right this minute, but like in a couple minutes. But,
I can try sharing my screen,
okay.
and if it doesn't work, then I need you to run it on your side.
Yeah.
I'd like to try sharing my screen. I suspect I'm supposed to share my screen this afternoon because I have a committee meeting. So we have to decide what I'm doing for my, what's it called? PhD dissertation
Committee, yeah.
thing,
Usually,
whatever.
yeah.
Candidacy exam, that's it.
Oh, okay, yeah, candidacy exam, yeah. It's usually something like that. It's
I have to do a candidacy exam, and it's a 6,000 to 8,000 word essay and a presentation. But it needs to be about a project, so it should be about something that I've done.
yeah.
And it needs to be publishable.
Okay.
So there has to be
yeah.
something normal about it. I'm not sure. They can, if they want to decide what I'm doing, that's fine, because I'm kind of out of ideas.
Yeah.
But I do have synchrotron data that hasn't been used. So maybe they'd like to put that in something.
Yeah, that would be great, because it's a pretty nice tool to have. Yeah, that sounds great. Well,
it's
yeah. Welcome to the meeting. Hello, Karan. It's the last meeting of the year. We'll take a break for a couple weeks and come back in January. And so I don't have a end of the year report because we did sort of a year summary in September. I did it for the OpenWorm annual meeting, and so that's sort of our recap for the year. Hello, Dick, and hi. And who is this? Hello, Jitster.
Veronica.
Is that the person is that the person who was invited?
Hi.
Hi, how are you?
Turn on.
Hi.
Hello. How are you? Would you like to introduce yourself?
I'm fine. Thanks.
Yeah.
Yes. Okay. I can hear you well.
Okay. Good.
Nice to meet you all.
Nice to meet you. Would you like to introduce yourself?
Yes, I am a PhD. I'm a biologist, and I worked on the mechanical design of sea urchin, the skeletal part. And I did a research on the structure of the echinoid test, that is the skeleton, to identify new biological insight regarding the structural organization, and also identify functional strategies to be transferred in engineering, civil engineering, industrial design products.
Oh, that sounds good.
It's a little bit confusing, but I hope it was quite clear. I don't know.
Oh no, it was good. Thank you.
Okay.
Yeah. We do a lot of that in this group.
She has a strong Italian accent.
Yeah.
Yeah. My English is so bad. I'm sorry.
No, it isn't bad at all.
No, it's fine. Well, welcome to the meetings, and we do a lot of this sort of stuff in the group here. We talk about like different approaches. We look at developmental biology, but also computer science and also some physics and engineering. And Susan's going to be presenting today on something like that. She's going to be presenting on soft materials. So this will be a fun presentation.
Susan is also an engineer like you.
Yeah.
Oh, right.
Electrical.
Hi.
I'm in biomedical engineering right now, trying to get my PhD in order.
Oh.
And I have a committee meeting this afternoon. They're going to give me my homework for 2 or 3 months.
So Susan, can you share your screen or try to?
I will try to share the screen.
All right.
Yes. No, it's grayed out. It won't share.
Is it a PowerPoint?
Yeah, I've got it here, but
Yeah,
she
Bradley
just
has
can't
it.
open
You
her
can run it.
Okay. You can do
screen.
the old-fashioned next slide.
Yeah.
Yeah.
All right, let me share my screen. Okay. And you can see my screen here.
Yes,
Okay.
yes.
All right, let me share the presentation. Oh, here we go. And there we go.
I'll just present, I think.
Yeah, go ahead. And then tell me when to change the slide.
Okay, this is a topic that I've been interested in for a long time. It's called active matter. And I guess next slide, this has to do with my thesis. I'm looking at tissue. And it's, viscoelasticity. So if tissue is alive, then it's actually active matter. Anyway, so active matter is made of units that consume energy or produce energy, and it occurs across time scales. I've got from galaxies to chemical level things like microtubules, and it's in a non-equilibrium state. It's a non-equilibrium thermodynamics. And this can even expand to small autonomous robots. Like you can actually make a system with small autonomous robots. I saw YouTube videos are very good for this, and I saw a YouTube video with the vacuum cleaners. They got a whole bunch of them, and they pushed around the boxes that they came in, and they made patterns. So they're kind of an active matter. That was kind of an active matter system. Okay, next slide. And here I've got it shown. There's a flock of birds, and that goes for flocks of sheep and flocks of human beings as well. And then you've got, these are microtubules, and they're in different concentrations of ATP. And then this is, the third one is 2 balls that are shaken up and down. And it's a very simple system, but they were able to really study, what did they study? Sorry. Reynolds number and the movement of small creatures through through water mostly. I think it was water. Next slide. yeah, here it is. And they have there's the fluid that they were in. They shook that up and down. And they found that this combination of balls initially moved backwards and then it moved forwards. So there was a crossover point, and that's what the critical onset transition was. And the plus signs are simulations. So they were able to get pretty good data and really nice simulations of what was going on. And so next slide, this is just fast. So you can see that the double ball, which they're calling a swimmer, initially moves down with a Reynolds number of 5. And then as it gets, as the Reynolds number increases, it moves forward. And on the graph, you can see that Reynolds number of 20 is kind of the transition point. So this is with a very simple system. They were able to study Reynolds number and how things move at low Reynolds number as compared to high Reynolds number. I thought it was really neat because it's such a simple system.
So what is the Reynolds number again? Just for people who might not know.
Reynolds number, it's the how fast fluid is flowing compared to the object you're looking at. It's you can
Susan, is this an oscillatory flow?
I don't know if we lost her. Let's see. Susan, are you still there? I think she's frozen. I don't know what happened. She's going to come back in. There we go. Back.
okay. Oh, next slide.
Susan, is this an oscillatory flow?
Yes.
Yeah.
And
Okay.
oh, sorry. It's shaking up and down.
Shaking up.
And yeah, and initially
Okay.
the dual ball system moves down, and then it moves back up. And actually, they've looked at some microorganisms, and they tend to move backwards and then forwards, just naturally.
Okay. So we
Brine
didn't.
shrimp, that's what they looked at.
Yeah. I think it cut off when you were explaining what the Reynolds number was.
Oh, sorry.
That's
Well,
okay.
it's just how fast the liquid's flowing, basically.
Okay.
And you get laminar flow at low Reynolds number and turbulent flow at high Reynolds number. That's kind of where I'm at with this. Maybe our resident civil engineer might be able to explain this better.
Yeah.
Probably. Anyways, this is just kind of a brief overview of this. So if you'd like to see more of this, about these swimmers in particular, there's a YouTube video there online about this. And some of these slides are actually from the YouTube video.
Oh, okay.
You can see that there's a little arrow there.
Oh, in which slide?
Okay, and this is the most recent. Yeah, this is the most recent from the most recent paper. Oh, sorry.
Okay.
Next slide. Yeah, it's
All right.
okay.
There we go.
And they've been looking at organisms in the intermediate Reynolds number and see how both sides of this influence their movement. So you've got low Reynolds number and you've got viscosity dominates, and then the high Reynolds number, it says inertial effects dominate. So in between, when you have all of these smaller organisms, you have influence from both. So they've been studying this intermediate Reynolds number using this double ball system.
Susan, where do fish go
Fish,
on scale? Yeah.
I would
Missing-
suspect that you've got small fish that would be more towards the viscosity end of things, and larger fish would be kind of like this, the dragonfly and the, I don't know what is, there's a snail there and I don't know. Is that an octopus or is that something else? That's a jellyfish.
Yeah. Yeah.
There's a curious report 50 years ago on zebrafish, which was made to swim down a channel with a thin layer of milk on the bottom, and movies were made. Okay, so the fish as a group actually set up whirlpools in the milk. And it looked like what the fish did was create a whirlpool and then use it as a peg to slip around. And they suspected that this reduced the drag on the fish.
Oh, okay. I know that octopus produce vortices that they use to push off of.
Oh, okay.
Yeah,
Okay,
that has some implications for heart development, I thought.
Oh,
Okay.
Because our heart beats, and it's actually a harder material in the middle of the embryo as it develops. I'd like to do some simulations on that because it's softer and then it gets harder and it pulsates. So
okay.
it probably affects things.
Presumably, most embryo development is a below Reynolds number, except maybe once the heart starts.
Oh, yeah.
Yeah, well, it certainly, and then it starts pumping blood, and then you've got a different system.
So is it the size of the organism here in this figure that sort of determines the Reynolds number regime, or is it the shape?
Yeah, it's got, traveling speed versus Reynolds number.
Yeah.
And yes, your smaller organisms are going through a high viscosity Reynolds number.
Okay.
Okay, do you know what kind of bird that is on the right?
No, I think it's just a bird. And
Oh, it's not a hawk.
maybe it's a starling. I don't know. It's just, you could probably put lots of birds right about there.
Okay.
Yeah. Okay.
Oh, it says it's a swallow.
Oh, okay.
Swallow.
Oh, sorry. I just taking it as generic bird here.
Oh, okay.
Anyway, they've actually done studies, I guess, on each of these. Okay, next slide. And you can, bacteria are also active matter. And they put some bacteria in with liquid crystals and got some interesting results. And they form a topology. It says bacteria mostly accumulate in the cores of positive topological defects, are expelled from the cores of negative defects. So, somebody in the audience, what exactly do they mean by positive cores? Is that like a hill? Anyway, I looked at this paper in a hurry last night to try to find that, and I didn't, but anyway, it's interesting. I also know that if you have pulsating flow to try to wash the bacteria out of cracks in, say, plastic material in a hospital, that they'll actually form a film and get stubborn and stay in the crack. So this is also a worthwhile study. Okay, next slide.
Okay.
Okay, and this is a bit busy, but depending on the actual shape of the cell, we'll have it be a kind of form of solid tissue, and if the cells change shape, they become fluid. And they've actually pinpointed this number as a Q point, where it's P divided by the square root of the area, and P is, what was that? I used to know that. Anyway, P is on the bottom here. They've got it as I think it had something to do with the vertices. So it's definition of the shape. And the V naught is the amount of movement.
Okay, if Q is dimensionless, P is perimeter.
Oh, maybe.
Because it would have the same dimensions as the square root of A.
Okay.
Yeah.
Yeah, it would. Thanks. Oh, well, next slide. I thought this was a less busy diagram. So there's your P again at 3.84, and you've got solid on one side and fluid on the other. And for the solid, the cortical tension dominates, and for the fluid side, the cell adhesion. Now, well, next slide. So these are the transitions in tissue when they actually move. It's more or less changing partners for the T1 transitions. That's the movement when they're elongating or doing something like what they call convergent extension. And then you have the T2 transitions, and one is cell apoptosis, which is the first one, and then the second one is a cell division. And when you have the apoptosis or the cell division, you can get rosettes, which is the next slide. There's some rosettes, and they tend to cause swirls in the tissue, and that tends to make the tissue more rigid in the area of the rosette. So this is important in development and in cancer development, as well as homeostasis, apparently. Like I have a quote about that. Anyway, these are all just sort of teasers as to what this whole subject is about. Some people spend their entire careers studying one aspect of this.
Yeah. So this is the tissue. Oh,
One last slide.
okay.
Sorry, what was that?
Oh, I was going to say, this is the tissue in this field, or is this like what does this field represent?
It's active matter, soft matter.
Okay, so the tissue changes its density or something like that as you go towards the slides on the right or the figures on the right. It's more concentrated. I mean, I don't know. I'm just asking or speculating. So it's like the.
I'll send you the paper. It's on rosettes.
Okay. Yeah. Okay. Tissue becomes more ridged around rosettes. I see. So these are kind of
Yeah.
like ridges and, okay.
Okay. Susan, I put another case for you in the chat.
Oh, we did. Another case? Okay. Anyway, the next slide is, this is the robots. And you can make small robots and you can make them into a swarm. And they say that this is not intelligent matter exactly, it's adaptive matter because someone external's controlling it. But you could probably make something that was sort of an autonomous system that was made up of small robots that moved and learned.
Right. I think we talked about this paper a little bit in one of the meetings. It's
Yes, you did.
where it's like the target shape and then the swarm would conform to the target and yeah. So that's good.
Anyways, I included this at the end because you presented it before, and this is actually active matter as well.
Yeah. Yeah.
So the external programmer should be regarded as another agent.
Yeah.
Maybe, yeah.
And is that agent intelligent?
Yeah.
Well, depends. Anyway, so I guess that's my presentation. It's just sort of a brief look at the subject, and not all thought out, but
Well, I think that's pretty good. Yeah. I thought there was a lot of good stuff in there, and I mean, some of it, we'll have to follow up on. A lot of good references in there. I saw at least 6 or 7 good references in there. Yeah. So this is Dick's paper that he put in here. Oh, this is the kinematics of explosively jerky diatom motility. So this is the paper here.
oh, okay.
Yeah.
Oh, I could have included that.
Yeah. Well, just saying.
Okay.
So did we have any questions, more general questions about this presentation?
Thank you for the presentation. I found very interesting the topological aspect, in particular the topological transition. I think that it's the slide 10. I don't know, maybe.
Let's see.
And also the glassy face diagram for tissue shape. The 8th one, the 8th.
8. This one.
Yeah.
Yeah. I didn't get the color. The colors. What are the colors associated to the different faces?
Well, the fluid phase is the yellow or orange.
Yeah, the other one in the B picture, picture B.
Oh, B. Yeah.
Yeah.
So this says it's instantaneous tissue snapshots. But if you look at the shapes, this is the fluid, and this is the solid, and you can see the difference in the shapes in the cells.
Oh.
And then this indicates their movement. Like they move a lot more. That each individual cell moves a lot more. That's its motion. Like each one of these colors is the motion of a cell.
Oh.
And
the interesting fact is that in the echinoid microstructure there is the same topological geometry there. So it's really interesting.
Okay, Susan, in the fluid case, do they actually change neighbors?
They change neighbors as in the diagram I had on 10. They do that shift in neighbors. If you show slide 10,
Oh,
yeah,
okay.
that's how they move. They don't come apart and start running around. That's not what they do. They shift. As in
Oh, okay. In transition one,
transition 1 mostly. Yeah,
mostly. Okay.
that's how they're moving.
It's interesting.
Yeah.
Thank you.
Can one do a random walk using transition one, or are you stuck in place?
No, you do a random walk.
You can.
You can. Well, you do that. You change your configuration, and then you do it again with another neighbor.
With another neighbor. Okay, so you can do it. Interesting.
Yeah. And then if you want to go in a circle or create a rosette, you have apoptosis occur or cell division. That's the way I understand it, anyways. Yeah.
Valentina, you know the word apoptosis.
What's the term? What's the term?
Apoptosis. It's cell death. Spontaneous cell death.
So
Okay. Okay.
The one in the middle, it shows a cell in yellow, and then, yeah, there it is. And then the one below it has no cell in the middle. The cell in the middle of the top part died.
Okay. Okay.
So then
This is why your fingers are separated, unlike the web of the duck.
Yeah.
they underwent apoptosis when you were very small.
Yeah, that's great.
I don't know what age in human that is, but
I don't know if I had any other questions. I like this slide, and this goes back to the liquid crystal biology discussion we had a number of months ago now, I think, where they use bacterial rods to look at their packings and how they move. And of course, they have these jamming phase transitions where they go from like moving in a fast, coordinated manner to getting stuck in place. And so, there's a lot of interesting stuff in this area that isn't really like tissue oriented, but it does describe sort of these how these collectives of cells move around and shape like.
you can get jamming transition. You can get jamming transition in cells.
In populations of cells with a certain density. So they're flowing through some current. It could be like liquid, and then they, at a certain density, they get jammed, and then they stop moving, and they have to orient themselves in a certain way. What was that?
Tissue.
Yeah.
Mesenchyme tissue.
Mesenchyme tissue, yeah. So I thought that was
That's
interesting.
an interesting one.
Yeah.
Bradley,
Yeah.
for my PhD thesis, over 50 years ago, part of what I did is what I call an Ising lattice membrane. In other words, it was a lattice system in which diffusion occurs across a narrow lattice. Okay?
Yeah.
And what happened as a result of this, there was a phase transition inside the lattice. And when the phase transition occurred, the flux across the membrane suddenly went down exponentially. Okay,
Wow.
so it was a system that switched itself off when you got to high flux.
Yeah.
No, I don't think I've ever read that. I'd love to see it.
It's an old paper, much older than you.
Yeah, that would be an interesting thing to. I don't know what is this your thesis or?
Yeah, part of my thesis, but I also published a paper on it. I'd have to dig it up.
Okay, yeah. Yeah, that would be great.
It should be in my paper, but it'll be a very early date.
Okay,
Around
yeah.
1968 or so.
Yeah. So that, like, when so I want to bring this back up because I don't know if we talked about it. We talked about it once, but this book, The Physics of Liquid Crystals by Desjardins, who was a French physicist. This was published in 1974. So this book sort of goes through a lot of the soft active matter stuff at a very early state. And this physics, of course, is useful in technology in building liquid crystal displays. And this is where you're using a certain approach to this. But then this is also very useful in biology, and especially like they've applied this to bacterial colonies and things like that. So this just kind of goes through all that was known at the time about liquid crystals, and he's a semi-theorist, so he kind of developed some theoretical techniques for this, which you'll find in a lot of the modern papers on this. So this kind of goes through a lot of different things. This is relevant, of course, to chemistry or to other types of systems where you have cells of different shapes. There's a emphasis on how cells are organized, whether they're organized, in one orientation versus another orientation. Let's see. From those sorts of orientations, you can find long and short range order. So he's kind of hitting on this theme of local versus global organization and how that plays into sort of the order of the whatever you're looking at. It could be a tissue, it could be an array of something, and so there's that theme, and then you can play around with what was that?
On that point, does he discuss what's called the wall effect?
I'm not sure. I'm just going through. Yeah, he talks about Landau free energy and some short order effects. So he does some statistical physics, but I don't know if he gets into the wall effect.
Because the thing that Susan mentioned about bacteria clinging pneumatically, if you try to wash them off, might be primarily a wall effect, where long particles line up against the wall preferentially.
Yeah.
Okay, even if they're totally passive.
I'm just taking a note here. I'll have to look. Yeah, I have to look at it. I should probably more effectively taken a short presentation on this myself and pulled some stuff out of the book. But I mean, I can send people this book if they're interested in going through it. It's more of a reference. Yeah.
Thank you.
Yeah.
Yeah, we don't.
I'm interested too.
Okay. Yeah.
I'll be interested. We would all be interested, it sounds like.
Yeah. So it kind of goes through the defects and textures and something called pneumatics, which is this ordering.
Oh, yeah, there it is.
Yeah.
Walls under the fields.
Yeah.
Okay, he considers walls. I don't know if he does the simple wall effect.
Yeah, walls associated with the Frederick's transition, and 180-degree walls under magnetic fields.
Might be there.
Yeah.
Okay.
And then pneumatodynamics. So he works out the equations of pneumatodynamics. So this is the dynamical state rather than just looking at it as a static system. And then cholesterics, which is where you get into a more complex geometry, and then smectics, which are liquid and solid layers with different orderings and all that. So that's this is like a classic book in the area, and it's dated now, of course, almost 50 years old, but it gives you a lot of sort of the basis for some of this, sort of the theoretical basis in that. So I'll send that out. It's kind of big, but I can.
My thesis, maybe.
Yeah.
Bradley, another question on this. Have you run across papers on pneumatic fluids into confined systems?
No.
No.
There's a bunch of papers about confined systems in the active matter material.
Oh, there is. I'd like to see them. Okay.
Okay. They confined actually some active particles in sound waves, and then they they let go of the sound waves and the particles just exploded out of the area. Okay, so you want confined active particles.
Well, or passive.
Yeah.
Okay.
Okay.
Yeah.
The reason I'm interested is the archaea, the polygonal archaea are extremely flat, which means they're a flat membrane back. So they're sort of all wall.
Yeah.
Oh well, can you see me any better now? No.
Oh, no.
You're right.
My camera isn't the greatest.
Yeah. Well, thanks, Susan. Oh, yeah. Thank you, Susan, for that presentation. That was very good. And, in the new year, we'll probably try to follow up on that a bit. And if people want to do something, be able to, we can maybe do some sort of simulation study. I know there are some other things that Dick mentioned about the archaea, so that might be something that we also tie into it, or we can just, I'm interested in seeing the archaea presentation, although I tried to contact Minock, and I couldn't get an answer from him, so.
He's been exact right now.
Yeah. So, we can maybe in the new year, we can get him to talk about that a little bit.
Yeah, University of Manitoba exams have been pushed back until, the 23rd or 24th of December.
We had six exams spread over about 3 weeks, I think.
Oh, yeah.
Oh, anyway, yeah.
So it's kind of useless for December, I think.
Yeah. So I'm going to transition a little bit. I wanted to talk about a couple things that are just news items that I found, or things I wanted to bring up. So the first of these is this simularium, which is a nice tool from the Allen Institute of Cell Science. So the Allen Institute, they've been working, this is one of the Microsoft billionaires, and they've given all this money to this institute, the Allen Institute. And they're trying to do a lot of stuff with using really high-end technology to find out more about the brain and about cells, sequencing the DNA and looking at the microstructure of the cells and putting this all into a digital format. So one of the big benefits of these what was that?
Yeah. Was that named after Bob Allen or somebody else?
Paul Allen.
Paul Allen.
Yeah. So so one of the upshots of this is they create a lot of data and they try to build these visualization tools. And so you can see some of these things here, but this is a platform that's in beta right now, Simularium. So this is something that you can actually share models. So you can build models around different biological processes and try them out in this platform. So they have these different types of things like actin and clathrin mediated endocytosis, and you have this sort of simulation model where it's like a 15-second shot of all the different components of this process. So this is written in CytoSim, which is a simulation language. They have Cytoscape. I don't think they're related, but there's this CytoSim. Yeah, so this is a software that they've developed, and this is the GitHub repository. So they have this CytoSim software that they're using to build this model, and then it's in this manuscript. So this is an actin and mammalian CME, whatever. Oh, this is this process here. So they have the model, it's open, and then here they have the visualization of the model, or a little sample of it. You can load your own data here. They have here's spatiotemporal oscillations in the E. coli MIN system. So this is where they have, I think this is written in Smolden. I'm not sure what that is, but this, Smolden 2.1. So this is from 2010. This is a platform that they have, the detailed simulations of cell biology with Smolden. So this is a very specific tool. I think this is also on GitHub, and then they also have the input data for it. So this is from a publication in PLOS Computational Biology. So this is where This is the MIN system, which is used to find the cell center during cell division. So they use this process, and they demonstrate it as the bacterial cell is turning in this space. So, I mean, you can do a lot of things. There are a lot of different software platforms here that they're archiving, and it's all open. So, you can add your own model or run from one of these models. And I'm not sure how hard it is to get some of these models to run. Even if they're open, it doesn't mean that they'll run well. So yeah, here's some more here on cellular biophysics. So this is also represented here.
Bradley,
Yeah.
let me make a proposal here. Go up, you see the one active.
Yeah, these.
Yeah, on the left. Okay, what I'd like to propose is that we use one of their software packages for a simulation of the cell state splitter.
Oh yeah.
Okay, because the cell state splitter, by my estimate, only has about 65 microtubules, for example, which means it's probably very statistical in a way.
Yeah.
Okay, so no one has ever tested whether it actually works.
Yeah. But it would be easier to do in a simulation, or at least you could try to.
Yes.
Yeah.
Yes.
Yeah, that would be
Okay.
great. Yeah.
Yeah, I might add that to the list of proposals to see if someone's I want to try this out.
All right. Yeah. Just making some notes here. Yeah, that's good. Yeah, I think that would be an interesting foray. And yeah, I think this is something called Medyan. I don't know. I mean, this is just, again, like the software is open and you can see the outputs.
Oh yeah, it takes some
Yeah.
investigation to get it running.
Yeah.
And try it in that configuration.
Yeah. So anyways, all right. So then the rest of so this kind of shows you kind of how this works. You have a JSON file, you load it in, you have this engine-specific trajectory file, you use it with a Python converter, or you can use a digital notebook. The simulation engine writes to the file format, and then this is based on this simulation engine, you get a specification file, you load it into a JSON file, and then you load that to a 3D viewport, which is on the website, so you can view your model there. So that's the way they envision this. So there's that. You can put it in the chat so you can access this.
Thank you.
Okay. And then there's also, the next thing I want to talk about is this paper, or is a Scientific American article from, I think, today or yesterday. This is actually on Mike Levin's work. Again, we've talked about Mike Levin and Raphael Yuste, who's their neurotechnologist. It's called New Clues About the Origins of Biological Intelligence. So, common solution is emerging in two different fields, developmental biology and neuroscience. So this is, Mike Levin does this work with sort of developmental systems and non-neuronal cognition, and also that, just regular cells, not just neurons, have electrical potentials. Maybe not action potentials, but they have this electrical activity that contributes to regeneration and developmental organization and things like that. So, they kind of write up this this is a popular science article. So they kind of propose that intelligence is a purposeful response to available information, often anticipating the future. This is distributed throughout biology at many different spatial and temporal scales. So this means that, they're kind of expanding this definition of intelligence. I think Mike Levin also wrote a paper with Daniel Dennett about something similar to this. And it's kind of an argument for sort of intelligent, that's ubiquitous. And so, we think of like people or mammals or birds, certain species that are intelligent, if they can use tools or if they can do, puzzles or something. But what they're arguing is that intelligence is much more basic. and it's this intelligent, purposeful problem solving. So you see this in cell tissue, single cells and tissues, in neurons and networks of neurons, in viruses and ribosomes and RNA fragments, and so forth. So you see this at different scales. And so they propose that the origin of intelligence is the central problem in biology. So this is probably a pretty controversial view, but the way they're casting the net here is maybe a bit provocative, but there are probably a lot of interesting questions surrounding this idea that there's this sort of behavior that's coordinated, and then how do you propose this? What are the sort of the driving mechanisms of it? Are cells autonomous agents, or are proteins autonomous agents, or what is the level of sort of autonomy that contributes to like if cells are autonomous, do they use that autonomy and coordinate their behavior to create tissues, and then those tissues are coordinated into an organism and things like that. So it's really kind of opens up a number of interesting questions. So these are things that, again, like this gets pretty philosophical, but there are a lot of interesting experiments one could do. But I think it's interesting that it's kind of like focusing on this common solution from two different fields, developmental biology and neuroscience. So, you have a lot of things that are going on in parallel in these fields. There's three parts to this argument. The first one is modularity. So natural selection is looking for, good designs, I guess, for things. They allow the organism to survive, it's fit, these sorts of things. And this is all modularity because these modules will form an evolution and they'll tend to be conserved. We talked about Hox genes and how Hox genes are conserved in their collinearity and their function. So, you get these different segments in an organism that get conserved. You also have, you have this thing about modules. You also have hierarchies. So you have lower level modules forming higher level modules and so forth. And then finally, the third thing is, down to the end here. Not sure they get to the third. Well, they were talking about hierarchy a lot. But I don't know what the third thing is in this list. Pattern completion. So pattern completion is where activation of part of the system turns on the entire system. In our apartment building, the family would have one central figure, let's say one of the parents, who would represent the families in meetings and engaged when it's needed. So, like one member of the group would go out and interact with the world and come back and maybe get a consensus amongst the members. And so that one individual thing or person or component could communicate with other parts of the network and then come back to that little part of that module and, sort of get a consensus view of things and then transmit that information back to the rest of the network. So this is kind of the way they view this.
Two comments.
Yeah.
One is the idea that morphogenesis and intelligence are related was spelled out about 70 years ago in books by Edmund Sinnott. Put his name in the chat.
Right.
Okay. This may be an advance on his ideas. They're not new.
Yeah.
Okay?
Yeah, I think so. Yeah.
Okay.
Yeah. And so yeah,
And
Edmund's
question about pattern completion. Does pattern completion have anything to do with the visual fill-ins?
yeah, there is that aspect of it in vision where you have
It does. Okay.
yeah. So you have yeah, in a visual system, you'll get like these lower level they'll get lower level processing of images, and then that'll lay out the pattern. But then if there's something that's missing, if it's an ambiguous pattern or something, then there are higher up processes that'll fill in the gaps in that. And so they've shown this in a lot of psychology experiments where they'll give people a very ambiguous pattern.
We all have what's called a blind spot.
Yeah.
The phobia, I guess, or where the optic nerve meets the retina,
Yeah.
and yet we're not aware of it because we told that in.
Yeah.
Okay.
Yeah. Okay. And I put the link to this article in the chat as well. Finally,
Thank you.
Oh, yeah, you're welcome. Finally, this is for people who are interested in machine learning, maybe more, but there's some interesting papers. So this NeurIPS conference is going on or it went on last week. So there are a lot of different, this is a huge conference on machine learning and deep learning. And so I know that Karan is probably pretty interested in this, but this is the top 10 papers and then the best papers of the conference. So I'll put this in the chat. And this is, so in this article, they give their opinion about some of maybe the best papers at the conference. This is the number of papers that are published at this conference over time. So it's really grown over the last 10 years quite a bit. And so a lot of these papers are on archive. There are a lot of different topics that are being explored in the community right now. There are things like gradient starvation, which is a way to look at like different ways neural networks use gradient descent and how to improve upon that process. You have uncertainty quantification. You have some issues related to newer models. They are always proposing different new models for doing this. So they have some pre-trained models for language. They have variational Bayes reinforcement, which is Bayesian techniques for predicting patterns or predicting categories. Contrastive losses, MLP architectures, which are a special new approach. I don't remember what MLP means, but these are some of the newer methods. Revisiting ResNets, so people are innovating in old models. And then there's some good papers in here where you have the why they did this paper and the key insights. So this is a 3D generative model for structure-based drug design. So, this is a model where they're using BioML, and they're looking at different generative models applied to drug design based on protein bindings. This one here, the emergence of objectness learnings. So this is segmentation from video data. So we do talk about that a lot in our meetings. This is humans can easily track objects for which you've never seen and not recognized. Machines should do the same. So this is the notion of objectness. So they kind of get into what an object is and kind of working on this from a technical standpoint. So they use a combination of single frame reconstruction loss, which is a segmentation network, and then a motion network that sort of takes video and segments the images and then looks at the motion cues and then kind of integrates that. So they use a reconstruction and a motion map, and then they're able to do these self-supervised segmentations in the paper. So, and then so there are a number of other ones: language models, retrieval models. Again, more stuff with video, actually multimodal processing, multimedia processing, the VATT model. So this is something they call a transformer, and this is for self-supervised learning of raw video, audio, and text. And this is this is looking at multimodal approaches to taking apart videos and finding features in them. So we usually talk about microscopy images, but you also have these other types of data that might be useful in other types of videos. And so I don't know if we have an equivalent in looking at like biological images. I'm sure there's a lot of auxiliary information that we could extract using sort of a multimodal approach. But so yeah, there's a lot more here. I could go through all day probably. Here's Karan. Hello, Karan.
Yeah. Hi. Yeah.
Thanks for attending the deck. Okay.
Yeah. I had some ideas. Like when it came to that intelligence, rating thing that was there.
Yeah.
how do you exactly classify that? Like when it comes to intelligence, do you have some sort of a metric for intelligence? Because I think it's like a random process is there. Okay, like if using that random process, be it a small organism, like there's a very basic implementation of that, like in the form of Conway's game of life. Like that thing is there, in which you have a very basic definition of what a cell is, and how that thing can grow. Similarly, when you have let's say a very complex system like the universe right now, right? So it's basically ways, structures, complex structures once they started to, form. So it's like a range of things that are there. Like if you talk about a particular species, what basically does is it has its own input-output mechanisms, like any other process. So it can gather inputs, in a lot of ways, and then it has limited processing power, the amount of neuronic matter that is there. So maybe the processing part can be quantified, in that scale, I think, but the input output matters and the amount of things that they have to classify and all that. I think I'm going to kind of going on a tangent here.
Yeah.
Maybe once I come to suggest, so, yeah. That is there, and I also wanted to show some more progress. I mean, with the axolotl later.
Oh yeah. That would be great.
Just a minute.
Thank you, Valentina, for attending.
Bye bye. Thank you very much. It was very interesting.
Oh, yeah.
Bye bye. See you
Bye.
next time.
See you next time.
Yeah.
Thank you.
Bye.
Bye. Okay, there you go.
Yeah. So the thing is, there's still a lot of noise, within the image, and I think I need to, I don't know, maybe I think if we have like, I was thinking maybe if if we had more like this is what I'm trying to do right now. I'm just trying to zero in on the actual object here,
Yeah.
and I'm trying to reduce the background noise that is there. So this process will take some more time because there's still a lot of noise that is still there to zero in on that. So if we had, let's say, can we get like images of this embryo in further developmental stages, like maybe till stage 3 or 4? Like I was going through this diagram here. So because the task is to map the development of the embryo and map the neural development of the embryo, right, from the outer surface, I think. Maybe something along these lines, because just this image, yeah.
That's a single cell, yes.
Yeah.
So I have other images, and I certainly could. Oops, is he there?
Yeah.
Yeah.
He's there.
Oh, you're there. Okay.
Okay, yeah.
Anyway, yeah, like I said, I really would like to get some more images this Christmas. Usually the salamanders lay this time of year, and I have that new microscope. So, hopefully, I'll get some nice images because I certainly
Okay.
Yeah.
yeah. And I can try to look up some more that are earlier stages where you can actually see the cells. That's an idea as well.
Okay.
So I'll try to look those up. Yeah.
Okay.
Yeah, as soon as I figure out what is happening with my, I have to write it. It's 6,000 to 8,000 word essay paper.
Yeah, you were mentioning it.
Yeah.
And I find out this afternoon. So then I'll kind of know better how I can squeeze that in there.
Yeah. So,
Okay.
yeah, I saw your notebook, and that looked very interesting. What are the new things you're doing with analysis? Looks like you had some new things.
Right now I'm just trying to, I'm just trying to, remove the noise. Then, I'm plotting the image, XY image, with the pixel depth here because of the, how do you call it? The value of the pixel that is there, right? As the z-axis. Something along those lines to get at least some ideas to how to go about plotting it. I could do like wireframes or surface plotting, This is just a scatter plot right now with points. So,
Okay.
just for a rough idea, how to clean more stuff out of the image. So I think this thing is still go on, but the thing is, because there are a lot of averages involved, right? because I'm getting rid of a lot of data within the processing itself. I had this doubt related to the, how to get that, like, what are the valuable inferences that I can gather, from that data, like, let's say with respect to neural development or something.
Yeah.
Or maybe is there some paper I can refer to, get some more insight into that aspect?
Yeah. So
Because
Susan has, well,
okay, I'm
she
not
has a
okay,
couple of,
okay.
oh, okay. Well, Susan has a couple papers on the technique that are kind of talk about the how the data is captured. So I mean, what's going on is you're getting this cell and it's rotating, and so you're able to see the surface of the cell. And if you had another
Yeah.
multiple cells, then you would be able to see sort of like how that process of cell division happens at a very early stage of the embryo. And then the thing about the axolotl is that you can see the brain; it's transparent. Like at the like if you were to see the thing in later stages of development, you'd be able to see like the brain as sort of transparent instead of being like kind
Okay.
of occluded. Yeah.
Well, early stage, it's not. It doesn't have a cover over it like Xenopus does. It doesn't have that layer of tissue on top. It's just it develops right on the surface.
Okay.
And that's why you can see it develop. But then it's opaque material does roll over and cover the whole thing. It does, but that's why I wanted to use infrared light to image it so I could see more of it, because you can see down into the tissue with the infrared light, but that's on hold, I think.
Okay.
But I'll try to get you some more images of
Images.
birds.
Yeah. Okay. Yeah, I mean,
So-
whenever you're done with the essay, I think this
Okay,
will
do I have your email address so I can send that to you? or-
yeah. Put it here.
Yeah. So yeah, that's good. And then I don't know if there are papers that he should read, like specific to axolotl development. I'm not really sure that I know that literature as well as you do, maybe. Or I mean, there's probably some like just basically a paper on the different stages of axolotl development, or I don't exactly know what level of granularity he would need, but just
I might take, what is it, Berchinsky's stages and send them to you as well, and maybe do an interpretation of what they say, because it's biological jargon. What is your background, Karan?
I'm currently doing my engineering. I'm in my third year. I'm majoring in electrical and computer engineering. So we have like courses from, let's say, DSP, signal processing, embedded systems, as well as software side, like machine learning, neural networks, and all that. Yeah.
Okay, so your biology, probably you don't want to do jargon in biology. It gets in the way.
It's like I keep on noting down, references, because there are a lot of things, that are thrown around. So
yeah.
Okay,
for
I'll try
future,
to
yeah,
come up with something for you. Okay,
like Hall's genes and cell state splitters and all that, all those things. Like I got the simulating part correctly. Like you could see the diagram that is there. And I could probably I'd like to start working on that model as well, because I think this could also be used for the axolotl data scatter plot, maybe in some. But
more images.
yeah,
That's
again, also we like relating to that Reynolds number thing, right? Like I think I had mentioned in the last thing about creating that viscoelastic model thing, right? But getting that tissue data set with the stress state diagrams, I went through papers, but I couldn't find anything conclusive, which could like give an inference about the maybe tissue shape, which gives an idea about that maybe. I'll keep on looking for that. because that's again something, I had another idea related to that if we could get these stress and strain, models for that tissue. Like there's another paper that I was working on with some of my these senior teachers. So she was mentioning, about quartz crystals and how, there's this problem in implantable medical devices, and how to, power those medical devices, we need like some other ways to power it, not just using electromagnetic induction and all those ways of powering it. So one of them was, using a tissue. Like there's some scientists that are using the heart as a way to, power their pacemakers and all that. So something similar maybe for the muscle area or something. So for that again, there's still a lot of things that I was trying to find, but I couldn't find something specific related to that.
Yeah, well,
That's still an active idea.
I think it's kind of a still developing area, because I'm working with optical quartz tomography, and we're trying to do elastography with it, like the viscoelasticity of tissue. And to do that, you have to perturb the tissue by pushing it in some way or pulling it, and then measure its change. And that's the optical coherence tomography would measure the change in the tissue when that occurred. So that's actually what I'm trying to work on, but I'm not working on axonal, partly because they're very difficult to image. You can see the blastocele develop, like that's the hollow portion in the middle. You can see that develop really well, and compare it to the tissue, that's fine. But to see the individual cells when it's developing is difficult. Like you can't, once they get to be fine enough, you just can't see the difference. It just looks the same. Can't tell the difference between cells. It's not like the fluorescent microscopy where you can actually put dye in between the cells. I've been trying to find a dye that works at the infrared in the infrared area, so maybe one could do that, but it's difficult. Like I found one that might work, but anyways, this isn't everything. This light goes through everything. All the different dyes, all melanin, anything you can think of. That infrared light doesn't think it exists. Just it goes through water, it goes through everything, which is a good thing. I mean, organisms have to live under the water, but that wavelength of light it's called a water window, and trying to find a diamond is very difficult. Yeah.
That's good.
Okay.
Yeah. Well, yeah. I guess so why don't we wrap it up for today, and why don't we get in touch? You can get in touch with Susan by email. She's not on the Slack.
Yeah.
Sorry, I put my email just quickly.
Yeah. And if you have any questions, you can contact me as well.
Sorry.
Okay, yeah.
All right. And so there you go.
If you just give it a second.
All right.
Did I get that right? Here's hoping. There it is.
There you go.
It's my email.com.
Yeah, and use this for,
Crawford-Young at gmail.com. So you can email me. It's fun.
And I'll post whatever, progress I've made, and as to what are the things I might need, for improving that model maybe.
Yeah, I'd like to improve the background image so it's not as noisy too. So oh, look, I had some with blue tape on it, and that seemed to work better from another person's point of view.
Okay.
So I'll try to find you some early stage with the blue tape. See if that helps.
Okay. Yeah. Okay.
Okay.
I can do that. Yeah.
Okay. All right.
Well, Thanks for attending. Talk to you in the new year, and we can
Yeah.
chat online if
Yeah.
you want. Okay. All right. Have a good holiday.
Okay.
Bye.
Bye.